Hello and welcome to part 2 of chapter 22, Origin of Species. So we've been talking about reproductive barriers, the pre-zygotic reproductive barriers. Now let's look at these post-zygotic barriers. Now, what does post-zygotic mean? Again, that just means a fertilization event has happened. So now you're starting to think about hybridization. All right, so there's three potential outcomes, or we can categorize the outcomes in these three ways, and we'll talk about each one here. The first one is reduced hybrid viability. So a fertilization event does happen, offspring is produced, but the offspring are less fit than either of the parental species. And the example here are these Ensatina salamanders, where these guys overlap and produce hybrids. The hybrids are kind of frail and weak and don't do very well, again, compared to the parental species. All right, so maybe you get a viable hybrid, like this example here with donkeys and horses crossing to create a mule here as an offspring. Here you've got a pretty hardy animal, but mules are sterile. So you have a sterile hybrid. So we've got a hybrid organism, but that's kind of a dead end from a biological perspective. Another possible outcome is hybrid breakdown. Now here, this is an interesting one because here initially, that first hybrid generation, first of all, it's fertile. So, okay, that's looking good. And you sometimes see something called what's called hybrid vigor. And it seems like, hey, things are going great here. These hybrids look better than the parental species. But that breaks down in the next generation, so we'll cross our hybrids again. And as we see here, these rice plants, here's the second generation hybrid. You thought it was going so well after one generation, and then by the second generation, the wheels come off. So those are some potential outcomes of these hybridization events. And these will continue to reinforce those reproductive barriers, right? Because you can't, even though you have reproduction occurring in these cases, you don't get really a hybrid that will give you a viable outcome for multiple generations. All right, so try this question. This combines a couple of ideas about speciation and hybridization. All right, yeah, the first part here, we've got a geographical barrier, so that's just allopatric speciation versus sympatric. And we have, if mating occurs, but the offspring eventually die out, that would be a case of hybrid breakdown. All right, so let's talk about more what happens when hybridization occurs, but in the context of these California salamanders, these Ensatina salamanders, which you've already heard a little bit about with hybridization. But this is just a wonderful example where we see speciation in a group of organisms that has occurred, and where we also see hybridization. And I'd encourage you to check out this video, go to the slides and click on this video. It's a YouTube video. It's only about 3-4 minutes long. Describing this work are the scientists that actually have done this. And you get to see some cool salamanders that they have. All right, so let's look briefly, though, at what's happening here. So if we look at the total distribution of these salamanders, whether we're, saying these are subspecies or potentially, are we really borderline on the fact that they are these species? This is sort of where we start to see it being difficult to make that distinction. We're like on the cusp of speciation here. And so the historical range for these salamanders would have been in the Northern California, Oregon, Washington, British Columbia. And these have over time migrated south and down through California, avoiding the San Joaquin Valley, not good habitat for these salamanders. But some populations have moved down the mountains of California and some moved down the coast. Those that live in these forested mountains developed camouflage coloration. So we see the body patterns here. Here's another picture of this, sort of showing more distinctively. Those that migrated down the coast here developed kind of warning coloration. Really, the coloration is a mimicry based on the fact that there are poisonous newts that have similar coloration in these same places. These salamanders aren't poisonous, but they look like those poisonous newts, so that, wards off predators. And so there's that kind of that speciation where we see different morphologically, very much differences between these organisms in these different locations. But the ring here comes back together for down the coast and down the Central Valley. The actual overlap where there's interbreeding is a little further south here. If I go back to this image, this dark area here is where hybridization occurs. And this was our example of reduced hybrid viability. So hybridization can occur between these much more camouflaged or warning coloration salamanders, producing a hybrid that isn't very good at either one of those things and has reduced viability. We can also look at the genetic differences between these salamanders. So if we cross the San Joaquin Valley here and make comparisons of these populations from, say, the coast to the northern part of the valley and work our way down, when we're making comparisons across the valley, there's greater genetic divergence. You could think of this like a percent, say 25%, 38%, 42%, 42, 44, 45. You see, as we get further and further south, that represents a longer and longer time that those populations have been separated. And so our genetic distances support these morphological differences. Whereas if we look along the coast here, we see these numbers being really small. So from here to here, there's only 3% difference. Up here, there is a big jump, but we would expect that because we categorize these guys as Xanthopicta, and the Schultzii are these ones indicated in brown. All right, so let's talk about these areas where hybridization occurs. It's called a hybrid zone. And this is where you've got those two different species overlapping and producing hybrid offspring. Hybrids often have reduced fitness, so that was the example of the Ensatina salamanders. But we'll see what could happen when hybridization occurs. There's a few different scenarios. But first, let's look at another really cool example of hybridization in a hybrid zone, which is with these Bombina toads. So these yellow-bellied toads, which sort of sounds like an insult, these guys are lowland toads. Got that wrong way around. These guys are upland toads. And the fire-bellied toad, these are the lowland toads. And so they tend to occupy different habitats, but there is overlap in their range here. And this is a fascinating hybrid zone. It's very narrow where they do overlap. And if we look genetically at, say, a marker, so we're looking at an allele that's typically found in our yellow-bellied toads, as we get closer and closer to that hybrid zone, it's still pretty high in our yellow-bellied toad, percent-wise. When you hit that hybrid zone, the frequency of that allele just tanks, and then you don't see it hardly at all in the fire-bellied toad, certainly once you get further away. So this is a very narrow hybrid zone. It's not some big overlapping area that's thousands of square kilometers. This is very narrow, but long. And if we look at this map here, we can overlay our cartoon map with a real map. And if we switch here, if we look, where do we see that dividing line? Why do we see the pattern like it is here? The Carpathian Mountains. So you've got those upland toads in the mountains and the lowland toads around the mountains. Go back. And so that's why you have this shape of a distribution. And we have this narrow hybridization zone. And we'll come back to these guys shortly. All right, so what can happen when hybridization occurs? There's three possible outcomes. And well, let's set it up first. So imagine we've got four populations here. There's gene flow amongst them. Then some barrier gene flow occurs, say, with this particular population of organisms, and we see divergence over time, but not so much that they can't interbreed anymore. So we say, well, they're different species, but they can still hybridize and produce these hybrid organisms in that hybrid zone when they come back in contact with these other populations. So what could happen? Reinforcement fusion, or stability. And let's talk about each one of those, starting with reinforcement. What are we reinforcing here? The hybridization is reinforcing the parental species. So as indicated in my little cartoon here, we see the hybrids kind of fading away. So hybridization is going to become less likely. We're actually reinforcing that these are parental species and distinct lineages of organisms. A really cool example of this is where we have organisms that have ranges that do overlap and don't overlap. So you've got the hybrid zone and then the non-hybrid zone. And so we can look in those different places. So where they overlap, these two different species, that they're occurring in symmetry, same place. And then we can look at what's happening where these species occur allopatrically. So species A is only found in one place and species B is only found in another place. They're not overlapping in those spots, but there is a place they do overlap. The example we'll use for this are these flycatchers, which are birds, and these two different species are found in Europe. And you have the pied flycatcher and the collared flycatcher. And notice as well, you have the collared flycatcher, say, where, in this blue range here, this is where you don't also find the pied flycatcher. So it's kind of like on its own in terms of flycatchers. Whereas, the pied flycatcher up here in the north, this is where you would only find it. But in this yellow area, you find both. So this is where both species overlap. This is potentially a hybrid zone, and where they occur in symmetry. And look at the difference morphologically. The pied flycatcher, these are the males, where there's no overlap. The pied flycatcher is kind of black and white like this, but where they do overlap, there's more of these brown looking variants of the pied flycatcher. And so this is really important when we think about mate selection, that these birds look a little bit different. Because look at the collared flycatcher, it's also black and white. And if we look at where the allopatric range is here, collared flycatcher down in the south, pied flycatcher in the north, these look pretty similar, right? So if, a female is choosing a mate based on appearance, that might be confusing because they look very similar. So where these species do overlap, we see selection for overt differences. The collared flycatcher still looks pretty similar. This is black and white looking appearance here where the overlap is. But the pied flycatcher, where overlap is happening, has this brown, these brown colored feathers. So it tends to look different. And so this is reinforcing that these should be separate species. The females are going to be less likely to choose these brown colored birds because they can, say, the collared flycatcher, they're going to be able to choose their own mates within their own species accurately because these look different. And so we can put this to the test where you've got males and females from of pied and collared flycatchers. And so let's just put them all together and see which males the females pick. Again, you got females of both species and males of both species. When you have the sympatric males only in our setup here, the females are really good at choosing their own species. So the pied females will pick the pied males, the collared females pick the collared males. We see all of the females are choosing their own species. If we set this up differently, so we take the females and give them the choice of these allopatric males. Now remember, the allopatric males of the pied and collared flycatchers, they look pretty similar. And the females aren't as good at picking their own species here. And so they do the majority of the time, but a lot of the time, a good chunk of the time here, eight of the females picked their non-specific male. All right, so wrap your head around that with this question and pick the best answer. All right, so yeah, this is asking which scenario is least likely. And if you took pied flycatchers here from the north and you brought them down to this hybrid area or further south, the females would have a harder time distinguishing those because they look like the males of the collared flycatcher, and therefore really hybridization would potentially increase. So the least likely choice here was that hybridization would decrease. All right, another potential outcome here would be fusion. This one's a little more straightforward. It's easy to conceptualize, I think, where you have two parental species, they come back together, form hybrids, and here the hybrid is as fit as the parental species or more, and you start to see a complete loss of those parental species and more of the hybrids. And so we're losing the parental species, and this is actually a way to potentially lose biodiversity because you've now lost these two species. They've fused into one. An example of this are our Lake Victoria cichlids again. This was a great example of where we saw sexual selection and speciation and diversification. But there's also a problem in, say, Lake Victoria and some of these other lakes where, due to pollution and other effects, the turbidity of the water, the sediment suspended in the water, actually inhibits the female's abilities to recognize those coloration patterns on their, say, appropriate male of their species. And so what we're actually seeing now because of this is a lot of hybridization between different species because of this inability to distinguish those different males. And so this is an example of fusion. Another possible outcome is stability, where you have both the parental species and the hybrids just continuing to be produced. That might mean that you have just this continuous input from the parental species and the hybrids are not very fit. If the hybrids do have increased fitness relative to the parental species, you might not see a fusion occurring because the hybrids are restricted to that one particular place. Just for example, they don't do well outside of that hybrid zone. And so you have this balance and then therefore continued formation of these hybrids and stability. And the example of that is one we've already talked about with our Bombina toads. So this really narrow hybrid zone, this just continues to happen. There's no evidence of these two species fusing. It's a huge geographic range. It's probably not going to happen anyway. There's not really a case of reinforcement. It would be a case of hybrid stability. All right, so what would be happening in a hybrid zone where reinforcement is occurring? All right, so yeah, where reinforcement is occurring, that's reinforcing the parental species and decreasing the persistence of hybrids over time. And so that means gene flow should be declining. All right, so let's look at now the time course of speciation. We can look at the fossil record for evidence of speciation and try to estimate, how long speciation might occur. We can use a molecular clock to estimate when species might have diverged. We talked about that in a previous chapter. And there's two general ideas that aren't necessarily mutually exclusive, which are gradualism. So we see this slow change over time, and we see these two lineages forming, and we might represent it on a tree like this with these long branches that stretch out. Another possibility is what's called punctuated equilibrium. And so what's happening here is you may have stretches of time where no change is happening and then really quick changes. And that's shown by these lateral branches. The change is happening so fast, like boom, now you've got two things here, another split, two things here. We have a bifurcation in our tree. So that's called punctuated equilibrium. And there's definitely evidence in the fossil record for this, where, again, you may have long periods where you're seeing some of the same species, and then there seems to be a transition, some sort of transitional event. And when I say that occurs rapidly, that still might mean millions of years, but we're talking about, geologic time here. And so you may have relatively these faster times of speciation that occur in the fossil record. Again, and that doesn't mean that this gradualism can't also occur. There may be a combination of both things that are happening. And so if we look at just an example of fossil record here, you maybe see, okay, there's something we would call stasis. So same species, same species, same species in a bunch of time in the fossil record. And then some pattern of rapid change where maybe we don't even see too many transitional fossils, or just because it's a short period of time, there's maybe not many opportunities to find those. And then you have another long period of stasis where you have these other different species that have arisen. All right, so let's connect that idea really back to this idea of phylogenies that we've talked about previously. What conclusion can we draw from this tree given the choices here? All right, yeah, this basically reinforces what I've already described. Is if we look at this tree, we have lineages that appear to have arisen through this punctuated equilibrium with these lateral lines here versus some of these other ones that appear to have arisen more gradually. So both things could occur. So it might be easy to imagine speciation being this sort of really gradual thing over time, but rapid speciation and how that might occur, it's maybe a little harder to conceive of if you're just sort of thinking about accumulation of mutations over time. So we talked about polyploidy before, and this is similar to this, where you can see a rapid change and speciation occurring. And the example here are these sunflowers, Helianthus anomalus, that arose through a hybridization event of two other species, but then didn't just stay as a hybrid, but rapidly diverged into a new species. And we can show this via laboratory studies as well. So those parental species are still around. And so you can take those and do a cross between them. And then over several generations, so you can breed those hybrids, they do just fine. And then you can dig in and look more closely at the genetics of this. So if we look at our sunflower species, H. anomalus here, and we look at the genetic markers, we find a mix of genetic markers that were from one parent species versus another parent species, and it's kind of mixed. The red and green are these indicators, markers from those parental species. So through recombination during meiosis, we see how these genetic markers have been mixed and combined, and ultimately we've produced very quickly a novel species. All right, a question for you then. The mechanism of macroevolution occurs by what? Right, so speciation, that's the core of macroevolution, and we think about it as at a big picture level. We're thinking multiple events of speciation. That's really how macroevolution is defined. And so these large-scale changes over time through, say, speciation, we can look at with an example here of diversification of birds over time. And so what you're seeing on this maybe complicated looking figure, but if we just focus on the red line here, that's the average line, the diversification rate, so think speciation. And why you've got these bounding areas around it, this just accounts for variability in the data. And so what we tend to see over time is increasing diversification of birds over time, so going from left to right, from 90 million years ago to the present day. Down here we have a relative extinction rate, increases here and there along the way for birds, but speciation overall has increased over time in the birds. And so these bigger type events, big speciation events, mass extinctions, that's what we'll talk about in the next chapter. I'll see you then.